Hello there. Good morning, good afternoon, good evening, depending on the part of the world you are listening to me from. Welcome to another lesson on the BED series, Biologists Explain This. I still remain your biology teacher, Peter Sabiodun, and today we continue our discussion on enzymes, and we'll be focusing in this lesson on enzyme inhibition. I'm sure you've heard about this concept inhibition. So, in today's lesson, we'll be talking about the different kinds of inhibitions and the consequences of inhibition. So, come along and let's enjoy this lesson together. So, let's start off by looking at our learning objectives for this lesson. At the end of this lesson, you will be able to describe competitive inhibition and also be able to describe non-competitive inhibition and be able to distinguish between these two kinds of inhibition, and lastly be able to use graphs to illustrate the types of inhibition and particularly be able to appropriately apply these concepts and principles you have learned to practical exam situations. And so let's begin by looking at what inhibition is all about. Now, when you talk about enzyme inhibition, you are referring to a process whereby certain substances called inhibitors bind to and then inactivate the enzyme catalytic activity. They bind to and they make the enzyme incapable of doing what it's supposed to do, meaning to catalyze the reaction. And so there are different kinds of inhibitors and they are specific in terms of what kind of enzyme molecule they inhibit or incapacitate. So you have the so-called competitive and non-competitive inhibitors. We're going to talk about them as we go in details. And then you have what they do. Okay, inhibitors, because there's this misconception about inhibitors. I mean, people think that it's bad news. Now, there are inhibitors created by nature to work in cells to control the rate of metabolic processes, because if you don't regulate metabolic processes, things will go on haphazardly in cells. Things will be disorderly. You can't have too much of anything. So what you have is that inhibition is a normal process of metabolism control in cells. But you see, man studies nature, man studies everything. What we learn in school are things that exist in nature, and then we make them topics and put them in books for people to study and apply. So when man has understood the way inhibitors work in the body, whether plants or animals like that, man now creates all kinds of molecules that work by the principles of inhibition. Now, for example, herbicides, okay, pesticides, and then a lot of drugs, therapeutic drugs used when somebody has an ailment or a disease or a disorder. A couple of drugs like that work as inhibitors of certain enzymes to control the pathway and the person get healed. So the idea of inhibition is a natural phenomenon which is designed to control metabolic processes in the cell, but there is an adaptation based on what man has learned, and so man has created drugs and several substances to apply the principle of inhibition in different contexts. So that is the idea. So don't ever think that every time you hear about inhibition is bad news all the time. It's a part of control mechanism in cells. So now we begin by looking at competitive inhibitors, and that also means that we are looking at competitive inhibition. Now, when you are a competitive inhibitor, the first thing to remember is that such kind of inhibitors, they resemble the actual substrate. Remember, they are not identical, so they have a close structural resemblance. They look like it. We will see examples. They look like the actual substrate, either because they have the same functional group. Somehow they have a structural resemblance. That's what you should take away from this particular lesson. They look like it, they resemble it structurally, and that gives them the audacity or the right to compete with the substrate for the active site. And when they pretend that this inhibitor, because what they do is they compete with the substrate for the active site and they end up occupying the active site, and then they prevent. So the effect of a competitive inhibition is that the inhibitor prevents the actual substrate from being able to fit into its active site. And so what it means is that if the substrate can't fit in, ES complexes will be few Okay, there will not be as many, and so you are going, instead of having ES complexes, you end up having EI, enzyme inhibitor complexes, and this will not definitely lead to product formation. And so, but the good news is that if you increased the substrate concentration, you can undo this kind of inhibition. Simply put, competitive inhibition is usually reversible. Let me say that again for the sake of emphasis. Competitive inhibition is reversible. How? By simply increasing the substrate concentration. In other words, the molecules of the substrate, they will outcompete the molecules of the inhibitors, and that will undo, okay, the competitive inhibition that we have been discussing so far. Just increase. So by simply increasing the substrate concentration, it can be undone. And let's look at examples of competitive inhibitors. Now, if you look at bacteria, they have a cell wall made up of peptidoglycan, and to make this cell wall, okay, bacterial cells, they need folic acid, okay, hydrofolic acid, that's the full name, and dihydrofolic acid. And it's what you have is that they get folic acid, that is this dihydrofolic acid, they get it from para-aminobenzoic acid, PABA. So the procedure is this: if you need to convert PABA to dihydrofolic acid, that's the target of some pharmacists, some drugs called sulfur drugs. One example is sulfur methoxazole. What these drugs do is that they are structural analogs. They are like generic drugs, so they look like PABA. And so what they do is that they compete, okay, they act as inhibitors that will bind to the enzyme that is required to convert PABA to folic acid that bacterial cells need for their wall. And so what they do is this synthase enzyme that is required, this dihydropterate synthase enzyme required by bacteria to convert PABA to hydrofolic acid, that is what is inhibited. So that's a picture of a competitive inhibitor instance. So look at this diagram to just simply illustrate the concept of competitive inhibition. Look at succinate, for example, and look at malonate. Now, if you look at them, the difference between the two of them is just CH2. Okay. Now, succinate has two, okay, of those groups, and then malonate has just one. So you can say that malonate resembles now the enzyme that is supposed to act on this succinate, okay, it requires this molecule as a substrate. But malonate resembling it can act as a competitive inhibitor. If you look at it, no product is formed. Look at the diagram below in blue. The one at the top, the actual reaction is in purple up there. So you have an example. So some succinate is supposed to be converted to fumarate, but because of malonate pretending to be like it, there's no reaction that results. Let's turn our attention now to non-competitive inhibitors. Now, this kind of inhibitors, from the name, they do not have any structural resemblance to the actual substrate. So they don't compete with it for the active site. Instead, this kind of inhibitors, called non-competitive inhibitors, they bind to another site on the same enzyme protein molecule. This site is called allosteric site. And so what they do is that they bind to this site and they distort the shape of the active site. Of course, once the active site is distorted, it's no longer complementary to the substrate molecule, and that means that no reaction results. And so what is important is that there's a change in the, so you have a conformational change to the active site, so that the active site is no longer complementary to the substrate molecule. There will be no ES complex formed, hence no product formation because there's no catalysis. And still on non-competitive inhibitors acting, they lower the rate of the reaction by reducing the amount of active enzymes. Remember, just imagine all the available protein molecules of the enzymes bound to their allosteric regions by different non-competitive inhibitors. So that means that you are destroying a variety of active sites that are supposed to be available for the actual substrate molecule to bind to, and that is the idea behind it. And you have an example of a non-competitive inhibitor there. Now, there's some metabolic poisons that you have that act on the respiratory pathway, in particular the electron transport chain that occurs on the mitochondrion. Now you have the last step of that process where electrons are being transferred down the electron transport chain. To transfer electrons to cytochrome oxidase, you need all kind of carriers. Now the last acceptor before oxygen is the cytochrome oxidase enzyme that also acts as an electron channel. So this particular enzyme is inhibited by metals like cyanide. Okay, cyanide, arsenate, they can inhibit. So what it means is that the transport chain that leads to chemiosmosis, so that you can have ATP synthesis, okay, that transfer of electrons is inhibited. So it's like the old channel is shut down. Electron transport chain on the mitochondrial inner membrane is shut down because of this poison, because they bind to the enzyme cytochrome oxidase. That enzyme catalyzes how water is formed. So hydrogen ion is supposed to combine with electrons to form hydrogen atom that will reduce oxygen to form water. That last step is what's inhibited. You also have arsenate that also inhibits that enzyme that you have as glyceraldehyde phosphate dehydrogenase. Glyceraldehyde is what is an intermediate molecule in the pathway of glycolysis. So you can imagine so many inhibitors. Remember, these are metals. They don't look like the amino acids at all. Okay, so they are not even organic. They are actually metals. So they are non-competitive, but their effects are far-reaching because they can distort the shape of the active site, and so there will be no catalysis in the long run. So study that graph. It's graphic enough, it's detailed enough, talking about the effect of a competitive inhibitor and a non-competitive inhibitor when they both bind to the protein molecule of the enzyme. And let's still on the comparison. Look at this diagram. You can see to your right, the inhibitor that is non-competitive is colored yellow, binding to the allosteric site. So you can see inhibitors on both sides, in the competitive instance and the one that is non-competitive. In the enzyme A, the substrate could not bind to because the inhibitor is occupying the position meant for it. The other one, the inhibitor that is called non-competitive is binding to the allosteric site and has succeeded in distorting the shape of the active site. So the substrate molecule could not. In both cases, there was no catalysis, no ES complexes, or let's say fewer ES. But for the exam, you have to be very careful. So it's better to use the expression fewer because there will still be some cases where the reaction will still occur. Not all enzymes might have been affected. So the safe thing is to say that fewer ES complexes are formed. Avoid saying no ES complex because it's possible that some enzymes are not affected by the inhibitor. So please note that it's safer and more academic to talk about fewer ES complexes are formed. So that means that the reaction is slower, not that it will not occur. There will still be a reaction, but it will be very slow, and the cell can't depend on that kind of catalysis. So comparison is still the same thing we are saying. Just look at the diagram, this diagram and the previous one on the other slide, so that I can have a clear picture of the way inhibitors affect the activities of an enzyme. So look at that table just for the sake of clarity and understanding. Study the table and I'm sure you understand where does it occur, for example, that is where does the inhibitor bind to, okay? And then is there any change? We'll discuss this later in the second lesson on inhibition. We are going to be discussing the effect of inhibitors on the Kmax, that is the Michaelis-Menten constant, okay? So the Km and the Vmax, okay, we will discuss what inhibitors do to affect the values of Km and then Vmax of an enzyme. So if you have questions on what we have discussed on this lesson, you can send your questions, your observations to my WhatsApp, my email, Instagram, and on the YouTube. I'll be very willing to respond to, and to answer you. Remember, we are working together for your success in your examination. I hope to hear from you soon next. If you enjoyed this lesson, tag your friends, share your experiences, let others also benefit from this lesson so that everyone can excel in this or examination. See you in the next lesson. Bye.